Welcome to Ask the Expert, and today we have with us two fantastic scientists from England. Alice Carr is a PhD student at the University of Exeter Medical School in the UK, working with Professor Richard Oram, Professor Sarah Richardson, and Dr. Beverly Shields. After a degree in natural sciences in 2008, which has enabled sorry, I had to let someone in, which has enabled a wide research field bridging traditional scientific boundaries, with her research exploring the interface between clinical science and islet biology. With a background in data science, she's also very interested in data obtained from the continuous glucose monitoring systems, or CGMs, and the use of this in understanding how variations in residual insulin production and other factors such as exercise influence the lived experience of type 1 diabetes. Having lived with type 1 diabetes for nearly 10 years herself, she's passionate about what research can do to make life better for people living with type 1 diabetes. So welcome, Alice. And we also have Sarah Richardson. She's an associate professor at the University of Exeter Medical School with a passion and interest in type 1 diabetes. She has been awarded 3 very prestigious fellowships from the Wellcome Trust, the Diabetes Research Wellness Foundation, and the JDRF 5-year Career Development Award. She was the first recipient of the network of NPOD, Junior Investigator Award, awarded for being a champion of collaborative spirit and data sharing for dedication to type 1 diabetes research. She co-leads the Islet Biology Exeter EBEX group with Professor Noel Morgan, and currently has nearly a £2 million funding, 1.2 million pounds as PI, and a co-director of 6 million research, £6 million Research England Expanding Excellence in England Diabetes Center of Excellence Infrastructure, patient pancreas samples. So it looks like they are building something absolutely phenomenal over there. It is centered around developing a clearer understanding of disease processes by which beta cells are targeted and destroyed. And she has particular interest in the role that enteroviruses and antiviral responses may play in the disease, and how age of diagnosis may impact, have an impact on diabetes endotypes. She curates the Exeter Archival Diabetes Biobank, which contains the most extensive and rarest collection of recent onset type 1 diabetes pancreatic globally. She's a member of the Tissue Prioritization Committee for the US-based network of pancreatic organ donors with diabetes, or NPOD, and she's been an innovator in the EDIA, diabetes participant in the Diabetes UK, and is the chair of the JDRF UK Scientific Advisory Council. She's also the member of Diabetes UK Research Studies Group, DRS G1, Cause of Diabetes, and is on the management board of the UK T1D Immunotherapy Consortium. She's an enthusiastic and proactive member of several international consortiums and has a real passion for team science. Wow. Thank you so much for all the work you're doing to push forward the understanding of the etiology of type 1 diabetes, both of you. And welcome. I'd love to hear, I guess, about what's going on at University of Exeter. And I don't know if Alice would like to share first. You're welcome to share your screen.
Yeah, for sure, I will do.
Also have to shout out to the pink you're wearing today. Thank you. That's Sugar Science favorite color, as you know.
Yeah, for sure, for sure. Can you guys see that okay?
Beautifully.
Yeah, perfect. Okay, yeah, thank you for the introduction. Yeah, as Monica said, I'm Alice, and I am going to take you through a little bit of the recent work that we've been doing in Exeter. There's been also joint with Oxford, so Rachel Besser in Oxford as well has been an integral part of this work. And it's some of the observations that we've been looking at with C-peptide decline in living children and adolescents with type 1 diabetes, and then also what we then observed in terms of beta cell presence when we've looked at the pancreas from donor children and adolescents too. And I don't know whether any of you actually know where Exeter is, but I thought I'd just give a little quick background as to where that is. But yeah, as Monica said, I've sort of completed my PhD, or I will have done on Thursday. My viva is actually on Thursday, in type 1
Fantastic.
diabetes.
Wow.
Yeah,
Big
and
day.
that's within, yeah, big day, big day. That's within the Diabetes Centre of Excellence as part of Exeter University, which is right down here in the southwest of England. And it is a beautiful part of the country. There are lots of stunning beaches, which might not align with your perception of England at all. And I do try and get out surfing quite as much as I sort of can. And there's also expansive moorland, which is great for sort of hikes.
It's
and
very
trail
beautiful
running.
there. My husband is English, so I've been there.
Yeah. Good. I'm glad you know. Yeah.
It's gorgeous.
Yeah, it's beautiful. And as you've sort of said, Monica, I think my my research experience is diverse. I've sort of said my PhD is in type 1 diabetes, and that's non-specific, I guess, for a reason. I'm a jack of all trades when it comes to research. I've trained in natural sciences, so I've got expertise in all scientific disciplines, sort of underpinned by data science and computing. And during my PhD, I've worked at the interface of beta cells and islet biology, all the way through to sort of whole living people. And that's trying to obtain better understanding of pathophysiology of type 1 diabetes using pancreatic tissue from the fantastic biobanks that we've got access to, mainly the network for pancreatic organ donors with diabetes, but also the Exeter Archival Diabetes Biobank, which is specific or curated by a man in Glasgow, but has moved down to Exeter now. And I work a lot also with continuous glucose monitoring data to get a better understanding of how biological factors like residual insulin production measured through C-peptide, and behavioral factors as well, like exercise impact glucose control. And I've also done some developing of classification models for diabetes. But yeah, that's the less interesting part of what I sort of was going to talk you through, just a little bit about me, so you kind of have a background as to what and who I am. But really the interesting sort of stuff is from what our recent study is, which, as I've said, was joint with Oxford. So Exeter and Oxford sort of working together. And they were, well, I'm going to talk you first through our observations of C-peptide decline in living children and young people across over a 10 years of duration that we've looked at, and then show you how this compares to beta cell loss in the pancreas donors across a similar duration length as well. And importantly and unique to this.
This is going to be very interesting, I'm certain.
Yeah, I hope so. I think unique to this body of work as well, we're able to look at these trends at different ages of diagnosis in children, from those diagnosed very young, so under the age of 7, through to those diagnosed over the age of 13. And so by stratifying our sort of results or our observations in this way, I'll then discuss perhaps then what that means for patients and clinical outcomes, and in the context of intervention trials, which I suppose is quite a hot topic at the moment.
It certainly is.
Yeah. So we all, I would hope everybody listening to this has some idea of what type 1 diabetes is, but if you don't, it's a disease defined by the inexorable autoimmune destruction of pancreatic beta cells, and that leads to endogenous insulin deficiency. And since about the 1970s, to assist with treatment decisions in both type 1 and type 2 diabetes and to monitor the course of disease, we've actually been measuring endogenous insulin secretion, but through a surrogate marker, and that's the 31 amino acid protein C-peptide, which is a byproduct of insulin biosynthesis, and it can actually be detected in the blood serum. And the reason that we use C-peptide over measuring insulin directly is because it's secreted in equal concentrations and at the same time to insulin, and it's got more favorable kinetics that allow measurement, and it's not affected by exogenous insulin, which is important. And again, since the sort of first detections of C-peptide in the blood serum in the 1970s, there has actually been a wealth of evidence that shows that a large proportion of people with type 1 diabetes do still have measurable circulating levels of C-peptide, and that demonstrates this persistence of insulin secretion. And in some cases, this is for many years after diagnosis.
Yeah, I mean, including medalists, right? I mean, some of these people have had it for the longest periods of time.
Yeah, so people that can see this graph sort of here, you've got people out here at 50 years duration, 70 years duration, that are still secreting, although small amounts, but measurable levels of insulin as measured through this stimulated C-peptide is on that graph. But we've also seen this from the pancreas. We've seen this in the pancreas. There's evidence from histological analyses from donor pancreata that show this persistence of insulin positive beta cells. And so really these findings together a long time ago now, but they challenged the dogma that all beta cells were destroyed at or soon after onset of type 1 diabetes. And that's sort of inspired a various number of sort of research studies trying to enable these beta cells to continue existing, perhaps against immune attack.
Yes.
I suppose also these sort of studies and all of this data that's built up, I guess, from the 70s, highlights that actually the autoimmune destruction of beta cells is markedly heterogeneous in people with type 1 diabetes, because some people experience more destruction perhaps than others, we could call it. And it's well known that there are differences in progression of disease between children and adults, but there is a recent body of work by P. Elite in Exeter and also others that has shown that even within children there are differences in progression, and that could be driven in what we think is an underlying endotype, so slightly different disease processes. And interestingly, these endotypes are strongly associated with age at diagnosis. So the studies that I'm referring to, it was seen it's sort of been looking at in longer duration in those diagnosed very young, so under the age of 7. They retained less C-peptide and also had less insulin producing beta cells, or islets that contained insulin producing beta cells, as compared to those diagnosed between 7 and 12, and even less as compared to those diagnosed at or over the age of 13. And this is in longer duration. This has been looked at.
Yeah, almost as if the immune system can't quite shut it down once it's established.
In the young,
yeah, as it gets, I mean, I heard a talk by David Harlan one time, and he was saying it's just the weirdest, strangest disease, because you expect the immune system to do its job, but it doesn't quite do its job, right? Because it doesn't quite shut all of the functionality down in some individuals. So it's very interesting.
Yeah, definitely. And so based on sort of the work, or and within this context of these recently reported potentially endotypes, these that associate strongly with age at diagnosis, we've sort of tried to look at what happens across duration, so longer, looking at this across the duration up to and over 10 years. And we first have looked at these trends in decline, as I've sort of mentioned, in C-peptide within living children. This is where it sort of started. And these were all diagnosed under the age of 16, taken, and samples were taken from the UK GRID cohort, which is one of the largest cohorts actually with random serum C-peptide measurements. That's 4,079 samples. And originally it was so large that originally it was set up for genetic studies. And then the next, we've looked at trends, or we've sort of compared then what we've seen in the C-peptide in the living people to the trends of beta cell loss within 2 of the largest pancreas biobanks that we've got access to, the NPOD and the Exeter Archival Diabetes Biobank as well. And actually, it's important for me to point out, so we have 235 samples there, but it's important for me to point out that an extensive analysis that can span duration from those diagnosed very young wouldn't actually be possible without the combination of these 2 biobanks. So it's this Exeter Archival Diabetes Biobank, which is sort of a key to what we're able to actually look at. And if I've sort of put this graph on here so you can kind of see that, but I'll talk you through it. As I've sort of said, we keep mentioning this Exeter Archival Diabetes Biobank. It's quite self-explanatory. It is held in Exeter, and it is archival. So it was curated around 50 years ago. And unfortunately, obviously, at the time when these biobanks were, this biobank was curated, clinical detection and treatment of diabetes wasn't as good as it is now. So there are lots of donors within the EADB biobank that were young and that died at diagnosis, which fills in a gap that we have with NPOD, the NPOD biobank, which is the more recently curated biobank. I think in 2007 it was curated.
Let me ask you, is this the first time that these 2 databases have been looked at together?
So there's been a little bit of looking at it with just at more of a cross-sectional sort of time point with a couple of the short duration samples,
Mm.
not as many. So we sort of put all of like this together, or more
Yeah,
of
you
these
really
samples together.
put the picture
Yeah,
together. And this is amazing.
yeah, for sure. So we were able to sort of look at, if you sort of look in this bottom left corner of this graph, you can see we've got a lot of samples that are young, that are diagnosed really, that have really short duration.
Yeah,
And so
as you
this
say,
EADB.
yeah, as you say, this is really filling in the blanks.
Yeah, to be able to sort of take it across the duration as well, not just looking at it at sort of one sort of time window.
Fantastic.
Sarah, go on.
You had a question?
Perhaps I was just going to add on there as well. We have done quite a lot of work of kind of trying to sort of see whether the findings that we have in the Exeter Archival Diabetes Biobank mirror what we see in NPOD in various studies that we've done over the years. So we do try to put the two together, but we've never done it on this scale before.
Yeah.
And as Alice says, looking in the, what's happening in both short and long duration disease.
Yeah, and then that's what I was sort of getting at. Like, I had not seen this kind of compilation before in the literature, and I think it's phenomenal.
Yeah, it's really important to sort of try and use what we've got. These NPOD's an absolutely fantastic resource, and we just don't have the short duration young donors.
Yes.
And that's a good thing, because that means people that are very young are not dying of diabetes at diagnosis.
That's true.
But we're using sort of this amazing biobank that was created by Alan Fowlis in Glasgow 50 years ago, maybe over now. And he put in so much work to take these autopsy samples and put it together and build this sort of biobank.
I think it's
and actually.
I was just going to say, I just think it's so important, like, to go back sometimes into either the literature or into these data sets and view them with current eyes, current scientific, wisdom. And I think that's kind of what you did. You really took advantage of going back into this, into this data set and brought it together with this, with the other more current one. And wow, I can't wait to see what else is in here.
I think as well, and Sarah, you can correct me if I'm wrong, but the EADB, a lot of the EADB samples are now like online for people to look at in the pancreatlas. Is that how we call it? I might pronounce it wrong.
No, it's right. That's pancreatlas. Yeah.
That's right. So you can sort of other people can look and try and use this really historical piece of data, I suppose.
Beautiful. Okay.
Yeah, cool. So I suppose let's get on to what we actually found, I suppose. So what did we observe in the data? First, I guess, starting with the C-peptide decline in the UK grid cohort. So we first, when we was looking within this C-peptide decline trends, we first started looking at proportions. So the proportions of children that retain detectable levels of C-peptide. I've put the graph on here, but for those people like just listening, across the x-axis, we've got categories of duration which we looked at, which is less than 1 year, so basically at duration 1 to 5 years, 5 to 10 years, and then greater than 10 years as well. And then on the y-axis is the proportion of those with detectable C-peptide, and then the coloured bars are the stratification of the age at diagnosis associated endotype categories that we've got. So red being very young, yellow is our in between group, the 7 to 12s, and blue is the over sort of 13 group. And what happens is, as you can sort of see from this, this isn't necessarily wildly new. We can see that the proportions of those retaining detectable levels of C-peptide declines across duration, and it happens for each age at diagnosis category. But what is the most striking here is this marked decline that we get after onset of type 1 diabetes in those diagnosed under 7. So you're dropping from nearly 90% or nearly everybody having detectable levels of C-peptide at diagnosis, and then after 1 year, far quicker than any of the other age groups, those diagnosed under 7 are dropping to 33% still proportionally retaining detectable levels of C-peptide. And so so this was really our sort of most striking sort of thing to note, was these young children seem to be rapidly progressing towards a near total or very, very minimal levels of detectable C-peptide. Very few people did. And so the next thing then we did when we sort of had found or seen this, we then actually looked at the absolute levels of detectable C-peptide, so what the actual levels were in those that retained the C-peptide. And we see this same pattern of decline again, but what is emphasized is that C-peptide is significantly lower at or soon after diagnosis in those diagnosed very young as compared to those diagnosed older. So as you can sort of see the red bars here starting much, much lower down in this under 1 year duration category. So those under 7 really starting low as compared to the 7 to 12 category and the over 13 category sort of up here. It's a huge difference. And it is also interesting to point out also on here that there is a group of people across all categories of age at diagnosis. So those diagnosed under 7, 7 to 12, and that should be 7 to 12, sorry on that, and over 13. At this 10-year time point, there is actually still a group of people that are retaining levels of C-peptide, and this is sort of interesting. It calls to an interesting line of discussion, perhaps we can pick up later. But there is still, no matter what age they're diagnosed, some people retaining still levels of C-peptide, detectable levels of C-peptide.
Fascinating.
And so next, taking from what we'd learned, I suppose, or seen in living children in C-peptide, what does it look like for patterns of or trends in beta cell loss? So these are from our pancreas donors, from the EADB and the NPOD biobanks combined, as we've sort of just talked about. And but I think first I'm going to just talk you through how we actually quantified beta cells. What do I mean when I'm talking about I've looked at C-peptide loss? How have I quantified this? Well, those that you're familiar perhaps with histological analysis, we've got to sort of identify islets in a pancreas that still contain insulin positive beta cells. And in the past, this has sort of been done by using light microscopy, immunostaining for insulin and glucagon, and then identifying islets that have got insulin staining in them. And actually for a small proportion of our samples, mainly from the Exeter archival diabetes biobank, because of their fragility and the rarity, we have to complete counts in this way. These counts are historical. But actually for the majority, we're able to interestingly digitize the slides and the samples using sort of this advanced technology called the Vectra Polaris and this halo analysis system. And the way that this works is it digitizes the slide almost as if you've taken a microscope view of it. And then algorithms within the halo analysis system can then identify different tissue areas. So I sort of put a little workflow of what this kind of looks like in the system. We've got blue is sort of stained, is tissue that's stained for glucagon, pink is tissue that's stained for insulin, and green is other tissue. And this is done through an algorithm when we tell it. And then we can then identify what an islet is based on some size parameters that we set. And I suppose just a close up of what this actually looks like. So you can sort of see the blue staining, the pink staining, the green staining, and then the islets that have been identified that we've set. We can then see and pick out which islets are still containing insulin or this pink stain, and those are the ones that are circled in blue here. Or we identify them as they're not containing any insulin.
Are these slides from a normal? Oh, sorry. Are these slides from a normal pancreas, or what are
these? These are from a type 1 diabetes pancreas. So so in a normal pancreas, it is unlikely you will find, and in fact, Sarah again can correct me, has anyone ever found insulin deficient or truly insulin deficient islets in a non-diabetic pancreas? I think to classify type 1 diabetes, we use a definition of they've got to have more than 10 insulin deficient islets, and almost in fact always in pancreases that have been identified as having type 1 diabetes, we will see that there is a near obliteration of islets across the pancreas. I don't know if Sarah, you want to weigh in on that, but
I mean, it's very rare to find insulin deficient islets in healthy controls. You do find occasional ones, but it is very rare, like far less than 1% of the islets that you would find in the section look like that. Whereas in a type 1 donor, it ranges from only, you know, a couple of percent to 60 or 70% of the islets that are still contain insulin. And obviously in people who've had disease for a long time, you might have no insulin containing islets left at all.
Do you see any distribution across the board in terms of the landscape of the pancreas? Do you see, you know, sort of loss of insulin in islets at the head, or the tail, at the center of the pancreas in different age groups?
So you frequently see lobular loss of, so you get sort of various lobes of the pancreas. You will often see lobular loss of the beta cells. And there is some evidence out there to suggest that you might kind of lose it from different regions of the pancreas faster than others. Yeah. But that's quite challenging for us to address in the EADB donors, because we only have 1 or 2 regions of the pancreas in there. But that's something that, you know, certainly can be looked at a bit more in NPOD donors. But yeah, there is some evidence to suggest that it's lost in certain regions of the pancreas faster.
Yeah. I had read that. I didn't know if your research also kind of supported that. All right, back to Alice.
No worries. Yeah, so these sort of digitized slides that we've got are enabling us to sort of pick, not only identify whether an islet's containing insulin or not, and that's, you can say whether a donor still contains or still has islets containing insulin producing beta cells. But we also sort of was then try to quantify sort of maybe how many beta cells are still there, and we've done this sort of by a surrogate measure of beta cell area. And the way that we've worked this out is we've done this as a ratio of insulin positive area, so that pink area that I was sort of saying from those slides on the Halo analysis platform, to the sum of the insulin positive and the glucagon positive area, so the sum of the blue and the pink. And so this is just this. I just put this in here just so this gives you a little bit of a context as to what I'm showing you, what I'm graphing in the next sort of couple of slides or throughout the next of the talk. So when we're looking at the pancreas. So just like before with C-peptide, we first started at looking at proportions. So proportions of our donors that retained islets with insulin positive beta cells, as we've identified through the light microscopy or the vector polaris system. And again, across the x-axis, we've got the categories of duration, which we looked across, and the y, these proportion of those with islets with insulin positive beta cells, and then the coloured bars are our stratification by the age of diagnosis endotype categories. And this decline again occurs across duration, again not surprisingly, for each age at diagnosis category, but there's this striking decline in those retaining insulin positive beta cells occurring in those diagnosed under 7 after 1 year duration, dropping from around 88%, which is similar to the start point of all the other ages of diagnosis groups, to after 1 year in the 1 to 5 year duration category, dropping to 8.3%, so 1 donor in 12. It's pretty marked in this graph.
Yeah, very,
you can see the decline.
Very striking.
Yeah. And again, so onto sort of our quantity, our surrogate for quantity of retained beta cells, which is measured through beta cell area on our digitized slides. We see this same pattern of decline. But again, what's emphasized is that this quantity of beta cells is significantly lower at or soon after diagnosis in those diagnosed very young compared to those diagnosed older. And it is important to note that we do get more variation in this graph, as you can probably tell, as opposed to our C-peptide graph that we plotted with the absolute levels. But this is because the samples are much smaller in these histological analyses. And although this is a limitation, it can't really be overcome because, as we've discussed, there are very few other samples, if any, that we can include in this analysis. Yeah. We're doing sort of the largest extensive analysis possible in child pancreas donors.
Yeah, no, this is very interesting.
What I'm going to do next now is just put these two observations from living and from donors next to each other, because this is what I also think is really interesting. That the trends that we're seeing seem to mirror one another. So on the left, we've got proportions of living children retaining detectable levels of C-peptide. And then on the right, we've got trends of pancreas donors retaining islets with these insulin positive beta cells. And we see this same decline across duration for all ages of diagnosis, this marked drop off in those diagnosed under the age of 7 after 1 year, and also a group over 10 years duration that still have some C-peptide left, and this group that in our donors that still have some islets containing insulin positive beta cells too. And if we look at the sort of absolutes, the quantity graphs, we do again see this same mirroring, but we see that those diagnosed under 7 are starting with less, so starting with lower levels of C-peptide or lower amounts perhaps of beta cells, as compared to those diagnosed at these older ages. So a little, I put like a little summary slide to try and that you could just maybe, if you have in your mind, this is something maybe to hold in your mind about what I guess the key take-home points of this are, is that we're getting this reduction in C-peptide secretion that mirrors the extent of beta cell loss across all ages and duration of diabetes within children that we've looked at. And personally, I think there's a very important point that our work highlights, or it's highlighted, I suppose, in the analysis that we've done, is that the children diagnosed under the age of 7 have lower levels of C-peptide and fewer islets containing insulin positive beta cells close to diagnosis. So I think those are the two sort of take home points. And I guess with those sort of points in mind, why is that? What does that mean? How is that important? Why is it clinically important? Yeah, exactly. Always a big question.
Yeah.
And we know sort of, so we've sort of talked about like this heterogeneity of type 1 diabetes in children, and we've talked how perhaps this is driven by underlying endotypes. Our work is supporting this sort of fairly recent and is possibly contentious hypothesis, but this data it's absolutely clear that there is differences in progression in children. I think that's the bottom line.
Yeah, despite people's kind of pushback on that as not a good business case. I mean, you can't just ignore reality.
I think it what we're showing is data, what we're showing is just what we're seeing,
Exactly.
And so it sort of goes with what's been suggested, or the endotype sort of hypothesis is sort of going with it. And I suppose that this rapid depletion that we're seeing in C-peptide and also beta cells mirrored in the donors as well, in those that are diagnosed under 7 years, in the very young, when we're just looking at diagnosis compared to after 1 year duration. To me, it really emphasizes that early intervention close to or before diagnosis is absolutely time critical in those progressing to disease in very early life, and it really calls for this necessity to improve prediction in type 1 diabetes. There is a couple of studies sort of that are recent that are again marching along this line as well, and I'd hope that this is sort of taken into account, or maybe these endotypes or sort of some of the data that we're showing is sort of maybe influence some of the maybe screening things that we're seeing.
Yeah, no, I think so. I mean, what you found here and what you're showing is certainly just supporting this whole idea that early screening, identification of those at risk, teplizumab, prevention bio, they're all trying to target that space. And I also feel like, it's becoming more evident, at least through the literature, that it looks as if there, there's a multiple hit model, which seems to be, kind of gaining more speed. And the fact that perhaps someone has a hit and then they go into remission, and then there's another hit and then they go into remission, right? And then that's the final thing to bring them to their diagnosis. So I think your work here really is great to illustrate the landscape of what's happening.
Yeah, and it's interesting you sort of bring up the teplizumab as well, like sort of pop that as the next and another sort of potential point to talk about these differences that we're seeing in progression, which we're observing in in this study. This is something that will influence or should influence intervention trials, and it potentially should be considered in planning and interpretation. It is worth noting that in the recent immunotherapy trials of teplizumab, people that were of sort of people at or pre-diagnosis of type 1 diabetes, all participants were actually over the age of 7. And with the suggestion that we're sort of saying or showing, and from others as well, that the youngest children are experiencing maybe a quicker progression to type 1, it's driven possibly by a different mechanism. One could speculate that if the effects demonstrated in the trials that we've seen would actually be replicated to the extent in children of younger ages. It is, the importance of these endotypes is something yet to be fully elucidated, and I know it's contentious. It's a nice debate topic. In Exeter, it isn't a debate topic. It is it's potentially gospel in Exeter, but no, that's a joke. But yeah, there's if we are, it is reasonable to sort of put out there that if we are to see an effective intervention or even a preventative therapeutic, younger children must be recruited into these trials to ensure efficacy.
Absolutely. I completely agree with this. And also, I just spoke with Jack Verosco, who's down at University of Texas, Austin. He does a lot of medical imaging. And he came, went to Georgia Tech, then he was at Vanderbilt with Al Powers, and then he's now at Texas. And he's looking at pancreatic volume actually declining during the first year after diagnosis, and he's really monitoring that. And so that's like also following along with what you guys are seeing, that not only like something is really changing morphologically, functionality, and those earlier patients should be included to really fully understand the spectrum of the disease. And I mean, while some might argue, endotypes, business case, the bottom line is breast cancer at one point was just treated with one broad brush, and now it's much more a personalized approach. And that's, my opinion is that that's where type 1 should probably follow, because it's doable, but you have to identify the different types of type 1 first. And I have a question in the audience from Braulio Marfil from Canada.
Yeah. How's it going?
Hey,
Hi,
Braulio.
Alice.
Hey, how are you? Good.
Great work. Great work. I really love what you did. And it really, like when you mentioned about the endotypes, I really was like, okay, I don't know the grid cohort that you use, but you mentioned something about genetic analysis that we're doing. So I'm wondering, do you have any data on HLA or maybe autoantibody status to actually look into this endotypes with your data?
So I don't have access to that genetic data. There has been a study that has looked at it with John Todd and Jamie Inshaw. So Jamie, I would note as well, Jamie Inshaw is another co-author on this piece of work that did try to look at different genetics, let's say, between across sort of those endotypes. Personally, it's difficult when you to Sarah's sort of I don't know whether Sarah you can weigh in on this, but it is difficult to try and separate out genetics in this way, and it kind of almost makes it it goes to the other argument which people are sort of opposing or try to oppose this endotype hypothesis with it, of that it potentially is a continuum, and I don't think that we're not necessarily saying it's not a continuum, but we're not sort of what's not being accounted for is the fact that those diagnosed under 7 have got some different mechanism of disease. I'm not sure how driven or that it is by the HLA, or how much we can pull out from HLA with that, as compared to those sort of diagnosed over the age of 13. There's some of the results in that study that they've done, which I think, is that published, Sarah? Is that, yeah, it's published.
Yeah.
I can't remember what journal it was published in. I was trying to remember where it was published in, but you could sort of have a look at that and take what you will from those results.
There are a couple of genes that they've identified that kind of pull out in the very young cohort, but it's not dramatic differences that they're seeing genetically, but it may be that it's something more to do with the time that you get the trigger and where the pancreas development is. So maybe it's not so much, you know, there is clearly some genetic components, but it might be to do with, so for example, those children diagnosed under 7 are likely to have got their first sort of evidence of islet autoimmunity, you know, kind of under the age of 2, if you look back at kind of natural history studies and stuff. And the stage that their pancreas is in development is very different to individuals who tend to get the disease a bit later. They tend to be the GAD, autoantibody positive individuals. They seroconvert that bit later in life, and the pancreas goes through this massive kind of remodeling and restructuring at that particular sort of stages of life. And maybe it's got something to do with when they get the trigger, what their pancreas looks like, how the immune system is evolving at that time, and all of those things come together to develop maybe a slightly different disease course. And it just seems to be much more aggressive in the younger children. And I think, as Alice has pointed out, you know, this has got really interesting implications for trials in the future. You know, obviously we need to make sure that the safety stuff is all done first in an older population, but hopefully we might be able to move some of these kind of immunotherapies into younger individuals, and we might hope that they're actually going to be more effective in them, because the immune attack seems to be more aggressive. You seem to get that more rapid destruction of the beta cells, which is what Alice has presented today.
Yeah, exactly.
Yeah, I mean, I guess you could say, if you're thinking about vaccinations, you know, I mean, children already, you know, experience, you know, many different vaccinations. So it wouldn't be, you know, out of the ordinary to think about using vaccinations on young children. I have a question for you. Great presentation. Very interesting data from Malin Floods from Tilburg. Congratulations. What is known about the C-peptide levels in different age groups among non-diabetic children and adolescents? Are they the same, or do they differ on average?
I wanted someone to ask me this question, because I'm really interested in it as well. I don't think this sort of extensive analysis kind of hasn't really been done, I think. And it's one key fact within diabetes research is we don't really know what's normal, really, or you know, we're looking at, sort of, Sarah's talking about how the pancreas is different, or might be different, and how sort of pancreas anatomy or morphology sort of changes as you go through, sort of as you go through puberty or you grow up, and so on. In what we do know, I suppose, most studies are done in we know from adults, I guess, or we look at sort of type 2 diabetes as well. People without diabetes have high levels of C-peptide, and when I say high levels, I talk in picomoles per liter, because that's kind of how we do it in the UK. But I'm talking into the thousands, and those diagnosed with diabetes are well, it's under 200 picomoles. If you have C-peptide over 200 picomoles at greater than 3 to 5 year duration, you don't have type 1 diabetes. There's something else going on. That's sort of the general sort of consensus. It was an interesting point, because it was something that I was thinking about. If we have different sort of remodeling of the pancreas, and maybe I don't know, again, Sarah, you might have to weigh in on this, about how the pancreas looks in younger children without diabetes, and how it changes, whether there's more islets. If there's more islets, are we going to get more C-peptide secreted or more insulin secreted? I don't know is the answer, and I would love if I'm missing some research somewhere, but it's absolutely something that needs to be done, because yes, we're making all these claims within type 1 diabetes, but yeah, I wouldn't expect it to look the same in people without diabetes, but it's something that certainly should be looked at and almost compared to, or we could almost normalize our data, or this sort of data too. Sarah, I don't know whether you.
Yeah, I mean, I would just say the same. I don't think it has been thoroughly researched, this. I don't know whether some of the information might come out from some of the natural history trials where they're following children and they can have, kind of the matched controls within there. But, not a lot is known. And in terms of, there's loads of changes going on within the pancreas at that point. The percentage islet area is higher in the children, but, relatively, it's still less than you would see in an adult, and we just don't know enough about it. Like you pointed out a minute ago, we don't understand normal brilliantly in terms of the pancreas and beta cell function and all things like that. So I think these are things that we really need to pick apart in order to be able to understand what's abnormal with type 1 diabetes. And so I think this is definitely an area of further research for everyone.
Yeah, it seems, wouldn't it be great? And if in addition to the CGMs, they would have, a continuous C-peptide monitor, right? You could wear one of those. That would be interesting as well. And really kind of get down to brass tacks with what that might look like in normals and those who are progressing to type 1.
I have to get Tim on that, Alice.
Our
dial up the biomedical
biochemist with absolutely.
Dial up the biochemical engineers.
It's interesting you just brought up sort of CGM, because I guess that was a sort of another point that I think is always quite is important to sort of note when we're talking about people with lots of C-peptide, people with diabetes with lots of C-peptide still, or people with diabetes that don't have like lots. We do see there's lots of studies out at the moment using CGM to try and sort of gauge a better insight, daily insights as to what's going on with people's glucose. And there are studies, and some of my studies as well, I've done, have sort of been looking at trying to the sort of relationships that we see with people with C-peptide from higher levels to lower levels within that, to see how that sort of impacts glycemic variability. And this roller coaster of diabetes is a roller coaster for everybody.
Oh, yes.
And so but actually these biological factors, so C-peptide actually has a huge role to play in how much of a roller coaster that roller coaster actually is. And so these people, these children, and in fact this goes for anybody that are not making lots of C-peptide, and in our study we're sort of showing that from diagnosis these kids don't have a lot of C-peptide. Really these children are going to perhaps experience a bit more of a roller coaster of type 1, and we should be really taking this into account in our clinical management strategies of them, earmarking these children or for more intensive management strategies.
That's an excellent
sort of closed loop and all of the systems that are out there, which I know, and in the UK there is the guidance out there that children diagnosed young do end up sort of going on to technology that's aiming to try and sort of ease this diabetes, ease the variability, the roller coaster of it, and the burden of diabetes. And that's sort of in guidance, but it really should be sort of the forefront of people's minds, is thinking this, or clinicians' minds. C-peptide does have an influence on diabetes. It's not the person and necessarily their management and their behavioral factors, which is the sole thing driving what clinicians might be seeing in the variability that they're observing. There is these biological factors that come into play. So that's quite a side divergent from CGM, but it just it sort of sparked me.
No, it's a great commentary, and together with Dr. D'Amelio at IU and another physician at Stanford, we are planning to bring up some of these points to a whole cohort of young physician scientists. Coming soon. So I think this work is beautiful. It really is. It's very carefully constructed. You guys really went through both at the tissue level, functional sort of, level in terms of C-peptide as a marker for function. And, I think it's going to bring, it's going to add more fuel to this, the hypothesis of there are definitely endotypes, and right, or whatever you want to call it, theory. Let's go with theory.
Theory, yeah. Let's do that. I don't want to upset people.
I know.
Yeah.
But anyway, I mean, it was a great talk. I so appreciate it. I wish you the best of luck on Thursday, and we can't wait to see what's next for you. Do you have something lined up next?
I possibly will be seeing Braulio a little bit more often. So potentially, investigating the pathway, or it's not that big news, but if the ADI, the Alberta Diabetes Institute, will have me, I will be likely heading out to Alberta to work a little bit more closely with islets, the islet transplant sort of patients, or sort of on that side of things. I know you had Braulio and James had talked recently to sugar science.
That's fantastic. Trained at a phenomenal institution and bringing the knowledge to another phenomenal institution to do more work. That's just absolutely fantastic.
Yeah, so we'll see. Yeah, thank you.
Well, keep an eye on what you're doing, Alice. Thank you again for talking with us. And
no worries.
We can't wait to share this talk with the scientific world. Have a great rest of your day, and good luck on Thursday.